Welcome back to the deep dive. Today, we're tackling something pretty fundamental. We're going deep into Chapter 12 of Gray's Anatomy to map out, well, the very first architectural plans for the human body.
It really is the origin story of our structure, isn't it? We're looking at that moment when a tiny, flat sheet of cells starts to plot out a three-dimensional person.
And our mission today is to help you visualize all of this, without needing a diagram in front of you.
Which is a challenge. And what's really amazing in mammals is just how late some of these fundamental rules get established. The axes that decide your head from your tail, your back from your front, that's not decided at fertilization.
Okay, let's unpack this. So we're at the blastocyst stage, and the inner cell mass has to decide which way is up. It almost feels like it's searching for its spot.
It kind of is. The future dorsal surface, so your back, usually ends up associated with the implantation site.
Right, where it gets the best access to the maternal blood supply.
Exactly. But there's still a debate about the mechanism. Is the trophectoderm just sticking to the wall and the inner cell mass has to align?
Or is the inner cell mass actively moving itself to get the best position?
Right. Either way, that location is what sets the stage for everything that follows. It's the first pin in the map.
So once that spot is defined, we have to start drawing the map itself. We need the three spatial axes.
Yep, the three orthogonal axes. You've got the main one, cephalocaudal, which is head to tail.
Then dorsoventral, back to front.
And laterolateral,
Yeah.
left to right. Plus, of course, the fourth axis, which is time.
And this is where the big transformation happens, right? Going from that flat disc to a folded embryo.
That's the key mechanical step. It's where 2D becomes 3D. If you picture that flat disc, there's a central oval, an ellipse. That part is destined to become the dorsal side of the embryo.
Your back, your spinal column, all of that.
All of that. And everything outside that central ellipse.
What happens to the periphery?
That folds inward. It tucks underneath to form the lateral and ventral structures. So the body walls, the belly. And the very, very edge of the disc eventually cinches together to form the umbilicus.
Wow. So a cell's starting position on that flat map literally dictates whether it becomes your spine or your stomach.
It does.
So here's the big question. How is this mapping actually done? What's the architect drawing these lines?
That responsibility falls pretty much squarely on the primitive streak. Its appearance is the event that confers the main axis, the cephalocaudal one. It defines back and, by extension, front.
But it's more than just a line, isn't it? It's also an engine for cells to move, a process called ingression.
Absolutely. And the path the cell takes as it moves through the streak is what defines its fate along that front-to-back axis, the dorsoventral one.
So it's not just that they move, but where and how they move through it.
Exactly. The cells that ingress and stay close to the midline, they become the dorsal structures, things like the notochord.
Okay, but how do they know to stay in the middle versus moving out to the sides?
Well, it's a very sophisticated funnel. You have all these complex molecular signals like BMPs and weights. Cells that enter at the very front of the streak are told to become, say, the notochord. But the cells that enter more to the side.
Like the lateral plate mesenchyme.
Right. They're given different instructions. They're told to keep migrating outwards, pushing towards the periphery. Their journey through the streak dictates their final address in the folded embryo.
That is just an incredibly elegant system. So that leaves the third axis, lateral, bilateral symmetry.
That one comes last, and at first everything is perfectly symmetrical. But that symmetry is broken pretty quickly.
As soon as things start to shift and curl?
Yep. As soon as structures from the splanchnopleuric mesenchyme, like the heart and the gut tube, start their looping and folding, you lose that symmetry.
And that gives us chirality, the reason our heart is on the left and our liver is on the right.
Exactly. And the source is really clear on why this matters. If you get the cephalocaudal axis wrong at the very beginning, you end up with major anomalies. That foundation is just so critical.
Okay, so the blueprint is locked in. Let's jump forward to stage 10, around the end of day 28. What does our embryo look like now?
Now it's something you'd recognize. It's a C-shaped embryo. The folding process is well underway.
And that folding has created the gut tube, right?
It has. The head fold has enclosed the front end, creating the foregut. The tail fold has done the same at the back end, forming the hindgut.
But the middle part is still open.
The midgut, yeah. It's still partly open to the yolk sac. Visually, at the head end, the forebrain is this massive projection. And just below it, you can see the cardiac prominence, that bulge where the heart is.
And what's happening with the scaffolding, the internal structure?
Oh, everything is segmenting. The paraxial mesenchyme is chopping itself up into these little blocks called somites.
And those are forming right next to the neural tube, which is zippering shut.
Exactly. And holding it all together, kind of like a central spine, is the notochord, sitting right between the neural tube above and the gut tube below.
And there are actual cavities forming now, spaces for the organs.
Yes, the intraembryonic calum is there. It's already divided into a central pericardial cavity for the heart, which connects to these two little canals,
The peritoneal canal.
the larger peritoneal cavities on either side.
So we have a beating heart in its own little bag, pumping blood. How reliant is the embryo on mom at this point?
Completely. The heart is beating, but it needs the chorionic circulation to be fully established, well, almost immediately. It's totally dependent on maternal blood for everything.
And externally, it's still connected to those other structures.
Right. It's in contact with three key vesicles. The amnion is draped over its back, the yolk sac is connected to its gut, and it's all floating in the much larger chorionic cavity.
Okay, that sets the stage beautifully. Let's drill down into the cells themselves, starting with the epithelial populations. These are the organized layers, the barriers.
They are the gatekeepers. And they all share this one core feature, polarity. They have a distinct top and a bottom. This lets them form tight junctions and create barriers, controlling what gets in and out of different compartments.
Let's start at the very top, the surface ectoderm.
Right, so that's the thin covering over the whole dorsal side.
Yeah.
This is basically the sorts for your skin. It'll eventually split into the temporary periderm and the permanent epidermis.
But it's not all just skin, is it? The text mentions these specialized zones, the ectodermal placodes.
The placodes are fascinating. They're basically patches of neurospecialized cells that decide not to join the central nervous system. They stay on the outside.
And they form our sensory organs.
Many of them, yes. One placode dips in to form Rattke's pouch, which becomes part of the pituitary gland. Others form the sensory lining for your nose, the lens of your eye, and the otic vesicles, which are the start of your entire inner ear.
And they also form the apical ectodermal ridge.
Which is absolutely critical for limb development. It's a command center that tells the arm and leg where to grow.
Okay, let's move a layer deeper to the neural ectoderm.
So this is the layer that rolls up and zips shut to form the neural tube. It's highly proliferative, and it gives rise to the entire central nervous system, brain, and spinal cord. And its edges.
The neural crests.
They peel off to form the peripheral nervous system. We'll get back to them.
And sitting right underneath the neural tube is the notochord.
The notochord is interesting. It's an epithelial tube, but it's non-proliferative. It doesn't grow. Its job is to be an inductor.
A signaling hub.
A major signaling hub.
Yeah.
It tells the ectoderm above it to become the neural tube. It tells the mesenchyme next to it to become somites. It's a project manager.
Got it. And the innermost lining, the endoderm.
That forms the epithelial lining of the entire primitive gut tube. Pharynx, respiratory tract, stomach, intestines, you name it.
Yeah.
But it also buds off to form the liver, the pancreas, the thyroid, and the lining of the bladder.
And finally, the coalomic epithelium, the lining of those body cavities.
Right. And this is often called a germinal epithelium because it's a source for so many different cells. It gives rise to the heart muscle, the connective tissues that support our organs, and of course the smooth linings of the pericardium, pleura, and peritoneum.
Okay, so if those are the structured, organized epithelia, let's switch gears to the other big category. The mesenchymal populations. These are the wanderers, the builders.
Exactly. Mesenchyme is all about freedom. These are individual cells moving through a 3D scaffold, the extracellular matrix, or ECM.
So the ECM is like a roadmap for these migrating cells.
It's precisely that. The stiffness of the matrix, the proteins in it, they all act as signposts, telling the cells where to go and when to stop.
Let's start at the midline with the axial mesenchyme.
This is a very specific group of cells right at the head end. When the embryo's head folds down, these cells get pushed out to the side, and they have one job.
Which is?
To form all of the extraocular muscles. Every muscle that moves your eyeball comes from this tiny population.
Incredible. Okay, moving out a bit, we have the paraxial mesenchyme, which we said forms the somites.
Right, the building blocks of the trunk. Each somite splits into three parts. First, the sclerotome. Those cells migrate inwards to surround the neural tube and notochord.
And they form the vertebrae, the axial skeleton.
Vertebrae, ribs, parts of the skull, yep. Then you have the myotome.
Myo for muscle.
Exactly. This forms all the voluntary skeletal muscles of the head, trunk, and limbs. And finally, the dermatome, which spreads out under the ectoderm to form the dermis, the deep layer of skin on your back.
Which explains the whole dermatome map that doctors use.
That's the embryonic origin of it.
Then we get to that really unique population you mentioned, the ones that break all the rules, the neural crest.
They are the great wanderers of the body. They never form a proper epithelium, and what they become depends entirely on where they start.
So, head versus trunk.
Big difference. The head neural crest is the sculptor of the face. It forms most of the connective tissues there. Cartilage, bones like the maxilla and mandible, ligaments, and even the smooth muscle in the big arteries coming off the heart.
Wow. And the trunk neural crest?
They take different paths. Some form the sensory neurons in the dorsal root ganglia. Others form the entire autonomic nervous system, sympathetic and parasympathetic ganglia. And a third group travels just under the skin.
And becomes...
the melanocytes. The pigment cells in our skin.
So the same family of cells that makes your jawbone also makes your skin pigment. That's wild.
It's an incredibly versatile cell type.
Okay, finally, let's hit the outermost mesenchymal layer, the lateral plate mesenchyme.
It splits into two layers. The outer one, the somites layer, is right under the ectoderm. It works with the apical ectodermal ridge to pattern the limbs. It also forms the bones of your arms, legs, and girdles.
And the inner layer.
The splanchnopleuric layer. That one wraps around the gut tube and forms all of its smooth muscle and connective tissue. It also plays a huge role in telling the endoderm what to become, like where to form intestines versus stomach.
We can't forget the little bit in the middle, the intermediate mesenchyme.
Right, that wedge between the somites and the lateral plate. It has a massive job.
Yeah.
It gives rise to the entire urogenital system, kidneys, gonads, and all the associated reproductive ducts.
And connecting everything, literally, is the angioblastic mesenchyme.
The source of all blood vessels. They form blood cells, the endothelial lining of vessels, everything. And it happens in two ways. First, by vasculogenesis, which is making tubes from scratch like the heart.
And then?
And later by angiogenesis, which is just sprouting new branches off the vessels you already have to fill out the whole network.
So what does this all mean? I mean, we've gone from the primitive streak, setting the body's coordinates, to the massive folding event that creates 3D space, and then all the way down to these specific cell populations, the epithelial barriers versus the mesenchymal builders. The level of precision is just, it's mind-boggling.
It is, and if we connect this to the bigger picture, one thing Chapter 12 points out is that while the basic genetic toolkit, the homeobox genes, are conserved across vertebrates.
Everyone uses the same instruction manual.
Right, but the timing of when things appear, what's called heterochrony, is really different between species. The schedule is flexible.
And that raises a really interesting question for you to think about. If the core genetic plan is so conserved, what does that flexibility and timing tell us about the evolutionary pressures on humans? We seem to prioritize things like forebrain development very early. What structures might have been fast-tracked in, say, a horse that needs to stand and run almost immediately after birth, compared to our own very long developmental period?
It just shows that evolution can tweak the developmental clock itself, not just the final product, to fit a species' needs.
You now have the fundamental spatial and cellular blueprint for the very start of organogenesis, a deep insight into the earliest moments of our own architecture. Thank you so much for joining us for this deep dive.